Thank you for the invitation. And I'm going to tell you a little bit on our effort to predictive oral mucosite to PROMOMIR, with the great hope that this research may contribute to care and cure cancer safely, as we all know that most of the ongoing cancer treatment cause side effects. And among the plethora of the side effects, we mostly focus on oral mucosite. It's an inflammation that mostly affects the oral cavity, the pharynx and the larynx of many cancer patients. You see a range between 15 and 40% of cancer patients are affected upon anti-cancer treatment by oral mucosite. And if we move it to a specific type of cancer, such as the head and neck cancer patients, which are mostly treated with the chemo and radiotherapy, almost 90% of these patients develop mucosite. This is, of course, a dual problem on the fact that part of these patients are hospitalized and part of these patients even has to interrupt the therapy and looking for additional treatment later on. Practically, there are no methods for diagnosis, early diagnosis of this oral mucosite, as well as there are no robust therapy that can be applied for oral mucosite. Most of the treatments are, I would say, empirical treatment, and initially there was a polyfermin that was a growth factor that was approved by the EMA initially, but later on was altered as this growth factor instigates aberrant proliferation. It was not something that cure oral mucosite. So our idea on which PROMOMIR is based is a sort of a combination between preclinical and clinical validation. We initially profile circulating microRNA from media of primary gingival cells that were treated with a broad range of anti-cancer drugs. And later on, we validated in patients treated with the chemo and radiotherapy, head and neck cancer patients. And we came up with a signature of a microRNA that was composed by 6 microRNAs. So this was tested in different platforms with different approach, and we found that the detection of this signature was quite flexible and easy with the different platforms. Then we start our procedure to patent this signature, these findings. Okay, so we initially start with Italy, and then we got the application number, then we moved to Europe, and then we made the step for the unitary European patent, and now we are expected in 2026, 2030 to be in China, in Canada, and in Australia. So the market in which oral mucosites are going to be involved is growingly expanding. That's for at least 2 reasons. We are expected 20 million new cases of cancer by the 2030. So this is already an issue. So more patients, more treatment, more side effect, more oral mucosite we are going to face in the coming years. And the other issue is that the most advanced therapy, including the ADCC conjugated, cause a severe bulk of oral mucosite. So all these new drugs that are coming into the cancer arena induce and cause oral mucosite. So where we are now? So now we are actively working in developing a multiplex assays that will allow to detect concomitantly all these 6 microRNA. So of course, we are going also to design clinical study, dedicated clinical studies to oral mucosite to validate further our assay, our signature. Of course, we are going to start a process of regulatory affairs and also to stabilize the marketing this kit, and of course, and hopeful being a public institution, the idea also to look for a spin-off consortium that could allow distribution of this subset. So meanwhile, we have started some collaboration due mostly to the interest directly, for instance, from AstraZeneca, which some of the advanced ACC conjugated drugs cause mucositis, with the Thermo Fisher, which they helped us, and we are co-developing these multiplex assays, and also with biotherapy and Aboca, which they are producing natural compounds, and natural compounds could be, at least in our opinion, the best way to treat or to attempt to treat a successful mucositis. We have also, I would say, institutional collaboration. First of all, with InnovUp, that is a large biomarker discovery platform that is supported by the Ministry of Health, and we are leading the liquid biopsy work package in such a context, so we can validate this assay across 17 different institutions, and also we are working with the other public grant and support that came from the PNRR. So this PROMOMIR was brought up originally by an idea of Dr. Sastrano, who looked to this, let's say, quality of life and circulating biomarker for quality of life, and later on was complemented by a number of young scientists that enthusiastically joined the research. And also, I wish to mention our technological transfer office and Emanuela and Giuseppe are here today, which of course helped us to develop. So we do hope that with the PROMOMIR, we can contribute to change or to reshape a little bit this path of the side effect in cancer that is understudied and say poorly known. We do not have therapy, and we hope to start this pathway that is going to look. And I'm here just to take your question and suggestions.